you can see the decrease over time when that stress was relieved. But you get this behavior at the whole leaf, but it's only this action at the guard cells that coincides with stomata closure. So as I said, abscisic acid has other has other roles to play, whatever it takes. It's not easy getting all of them. Has other roles to play. Okay. So if you look in Tice and Zeiger's book, before they had the two other co-authors, this plant this book, here's what you see, and it's essentially the same in 6 inches. During water stress, abscisic acid level in leaf increased 50-fold. In addition to closing, stomata have other roles to play in the plant, right? ABA and an alkalinization of the xylem sap are thought to be two chemical signals that the root sends to the shoot as the soil dries out. ABA closes stomata by causing long-term depolarization of the guard cell plasma membrane. What does that mean? When the environmental signal is given to the guard cell that's going to open stomata, remember the protons are pumped out, so you get a electrical potential difference across the plasma membrane and a pH difference. But that electrical potential difference is polarization. And when ABA is around, it depolarizes that. And I'll show you some of the putative mechanisms whereby that happens in a minute. But ABA depolarizes that. Depolarization believed to be caused by an increase in cytosolic calcium, as well as an alkalinization of the cytosol, so the pH inside the cell change. Increase in calcium inside the cell due to a combination of calcium uptake from the apoplast and release of calcium from internal stores, particularly in the vacuole. I think it's the vacuole. Anyway, this calcium increase leads to the opening of slow anion channels in the plasma membrane, which results in membrane depolarization. So potassium, the positive charge, moves out of the guard cell. Outward potassium channels open in response to membrane depolarization of rise pH, bring about massive potassium efflux. Okay, so, and this explanation is essentially unchanged a few years later in the most recent version. All right, so this change, all these changes happen, and this outward calcium efflux, right? Potassium concentration decrease in the guard cell. Okay. Let's have a closer look at what's going on. And don't worry about remembering details. Okay. As far as if you're worried about details and what you may have to remember, this is important. Okay. Remember this stuff. This stuff is interesting. And you'll see another one that's even more complicated than this that's interesting too. But I won't expect you to remember. Okay, but let's step through this. All right, ABA, this is a guard cell. Here's another guard cell in stomal aperture. Okay, so ABA comes around, however, comes to the guard cell. It binds with something that has an acronym, okay, inside the guard cell. This, what is our car? Abscisic acid binds cytosolic ABA receptors, whatever this thing is. They're not cytosolic ABA receptors. So it binds with them. that bound form of that thing, then right, it hooks up with this PP2C, which is a type 2C protein phosphatase, and it inhibits the function of that, right? That's what these sort of diagrams, are you guys familiar with that? That this means an inhibition, and this means a promotion. The arrow is a promotion, and this block thing means it inhibits. Anyway, that's like a biochemistry kind of a construct to use when you're drawing pictures like this. Anyway, it inhibits the normal functioning of this 2C protein phosphatase. Okay? This inhibition of this thing releases the positive regulator kinase OST1 from inhibition. So normally this thing inhibits this, but if this gets inhibited, this thing is able to function, right? And when this thing is able to function, it interacts with things in the plasma membrane, RBOH, which is a NADPH oxidase. So NADPH, remember, is a powerful reductant produced in photosynthesis. So it activates this, which makes reactive oxygen species in the apoplast, like hydrogen peroxide and this O2 dot minus, whatever that thing is. It's a superoxide, or I forget the name of that. Anyway, these things are nasty compounds, and they can inhibit lots of stuff. One of the things they inhibit is it opens up this calcium channel in the plasma membrane, which allows calcium to move from the apoplast into the cell. At the same time, this thing, OST1, this open stomatal 1 protein kinase, they call it, also promotes let's go this way. Let's do this first. This elevated level of calcium leads to activation of calcium-dependent protein kinases, CPKs, these things. These can also interact with this thing or this channel in the membrane, which is an anion channel, and promotes the release of anions from inside the cell to the outside, particularly chloride. Activated membrane. Okay. There are also these anion channels in the tonoplast, the membrane that surrounds the vacuole. So these anions, chloride, which tends to accumulate in the vacuole in guard cells when they open, that channel is stimulated so the calcium can get out here. Then it can get out of the cell, like that. This high calcium concentration also affects calcium channels in the tonoplast to release calcium from the vacuole into the cytoplasm, which just stimulates, it's a positive feedback. It stimulates even more and more of these type of channels that is a very effective and fast way of changing what's happening in the. All right, anion efflux. So losing this negative charge. Also is accompanied by opening potassium channels in the plasma membrane, so potassium can get out. And this change of, here's, okay, so all these things, if these words weren't up here, I couldn't explain it to you, right? But all these changes, here's what I can explain to you. This change in potassium and chloride causes the osmotic potential to change in the guard cell, get closer to zero. It's reducing the concentration of these little solutes. And that reduction in the water potential causes a disequilibrium between the water potential inside the cell and outside the cell. And water moves out of the cell into the apoplast, reducing the volume of the guard cell, reducing its pressure potential, and causing the small pore to close. You can read the rest of this stuff on that.
So the water moves out of the guard cells and then the stomata closes?
Yes. Yeah. So remember when the stomata are closed, the guard cell volume is small. When the stomata are open, the guard cell volume is large. When stomata are closed, the pressure potential is low. When stomata are open, the pressure potential is high, is positive, high. When guard cells are closed, their osmotic potential is relatively close to zero. Well, is closer to zero than when they are open. When they're open, their solute concentration is higher and their osmotic potential is more negative. Those are important things to remember about open and closed stomata and what happens in guard cells. Right? I mean, that's not beyond your capabilities to keep in mind. Right, it's a hint. All right. So and then when well, you can read this for yourself. Right? But you know, a lot of this stuff is still being worked out, and this picture in the next edition of the book will probably change. Right? But the important thing, and the reason I spend some time talking about it is not because of the details, but because abscisic acid sets in motion a series of reactions that ultimately results in potassium and chloride leaving the guard cell and its osmotic potential becoming less negative, and the stomata closed. The open stomata closed. ABA also inhibits extrusion of protons, and that's probably on this graph too, someplace. Yeah, here it is, right here. So this increase in calcium inside the guard cell inhibits this ATPase that normally would pump protons out of the guard cell. So increasing concentration of calcium act, as an inhibitor, and the pH change acts as an inhibitor of this thing, so that the protons don't get pumped out. So if the pore is already closed and ABA comes around, then these actions will not allow that first step to happen to open the stomata, namely pumping of protons across the plasma membrane. So there you have this last. So now let's go back over this just to beat the dead horse some more. During water stress, ABA level of a leaf can increase 54. In addition to closing stomata, ABA increases the hydraulic conductivity of the root. So if the abscisic acid is coming in the transpiration stream through the xylem up to the leaf, ABA makes that pathway less of a resistance to that path. So it increases hydraulic conductivity of the root. Increases root to shoot ratio at low water potential. It's okay. ABA and an alkalization of xylem sap are thought to be two chemical signals the root sends to the shoot as the soil dries. So here's the communication now between the stomata and what's happening below ground. AVA produced in cells in the root, gets dumped into the transpiration stream, that makes its way up to the leaf, makes its way to the guard cell, and they respond to that abscisic acid. Increased pH, okay, so maybe this pH change in the xylem has something to do with it. AVA closes stomata by causing long-term depolarization of guard cell plasma mirror. Okay, it lets chloride out, it lets potassium out, it inhibits the extrusion of protons. The membrane depolarizes, and so that inhibits the action of the ATPase that normally would pump protons. Increase, depolarization believed to cause an increase, is believed to be caused by an increase in cytosolic calcium, as well as alkalinization of the cytosol. So that's shown here. pH increase and this increase in calcium go together. Increase in calcium due to a combination of calcium uptake and release of calcium from internal stores. Okay, so that's shown on here by calcium coming out of the vacuole as well as calcium being taken up from the apoplast. In response to this channel, which is activated by these reactive species, which are produced by this thing, which is promoted by this thing, which normally is inhibited by this, but when AVA binds to it, it screws it up and that thing can go away. See this? Yeah. Okay. Where are we? Okay. Calcium increase leads to opening of slow ion channels, results in polarization. Outward potassium channels open, bring about. Okay, so I mean these words are summarized in this picture. It's not quite as simple as this though. And you know, this was state of the art in 2015 when we revised the book. Yeah, I don't want to go into it, but this paper from 2016 also shows these 14-3-3 proteins, whatever that means, some kind of protein, also interacts with all this stuff going on in the guard cells that's happening at the same time. It also is involved, these proteins are also involved in blue light signaling via phototropin in the guard cell. I talked about blue light and the xanthophyll zeaxanthin, but phototropin is also involved in blue light response, and that's mixed up with these 14-3-3 proteins. And they also inhibit all these things in the membrane that were on the other graph, but let's not go into this. If you like, you could read this caption to the figure, to this figure, as well as the paper itself. Okay, so it's a complicated thing that's going on in these guard cells. And since I don't expect you to remember this for exam or anything. It's why do I show it? It's a complicated scheme. It shows you that things are changing, right? Our understanding of the system is changing every year that goes by because we don't understand how it works. So people are discovering new things, and it's a complex system, but it's all coordinated. And one of the things I was struck with in reading through this subject material this year is how little attention is given to water potentials in these guard cells and outside the guard cells. Okay, so here, let me see if I can find it.